Welcome to Lecture 10 on the topic of genetic engineering. This lecture is part of the subject Future Farming Technologies, which is offered in the Bachelor of Agriculture and Technology. This degree is offered at both North Melbourne Institute of TAFE and Melbourne Polytechnic. Please visit our website www.nmit.edu.au for more information on this course and the other courses that we offer. My name is Dr. Nikki Cooley. This lecture will be separated into two parts. In part one, we will be looking at details and descriptions of different genetic engineering technologies. We will also explore some of the terminology associated with these technologies. Part two is going to be constructed of a series of case studies. These case studies, and along with their exercises, will introduce you to a number of the genetic engineering technologies. Please ensure you complete all associated exercises with this lecture, as this will complete your lecture notes. In this lecture, we will be covering two of the learning objectives. The first is to understand different technologies around genetic engineering methodologies in agriculture, and the second is to interpret, communicate and present ideas using the appropriate terminology for this particular industry. In part one of the lecture, we will specifically look at the transgenetics. We will describe some of these technologies, give example of both animal and plant transgenetics, and look at one agricultural case study on potatoes. We will also touch on recombinant technology. I would like you to pause your video here and watch the following YouTube link. This is a video that introduces you to genetic engineering. It concentrates on genetic engineering with bacteria. This is where this technology really started. Please watch the video, make some notes, and insert your notes here. Now that you have some understanding of the role of bacteria in genetic engineering, the following video is going to give you an overview of how that technology has been integrated into agriculture. Again, please pause the video here. Watch the YouTube video on genetic modification. Make some notes and insert your notes into your lecture notes here. So, let us start with the definition of transgenetics. Transgenetics is the process of introducing an exogenous gene, called a transgene, into a living organism so that the organism will exhibit a new property and transmit that property to its offspring. If I say this in more simple terms and not using the technical description of the definition, what I am describing is taking a gene that encodes for a trait that we want and put this gene into another organism where that trait can be expressed. When an organism has undergone a transgenetic, it is often called a genetically modified organism, and this you will see abbreviated to GMOs. The vector, or the way the gene is moved around from one organism to another, is a very important component of this technology. On this slide we have listed a number of vectors. There's the plasmid vector, the liposome, the viral vectors, the pronuclear injection, the protoplast or stomatic fusion, and the ballistic DNA injection. All of these technologies or techniques have the same aim, that is, to remove a piece of DNA and enter it into another organism so that it will enable the expression of the trait that you are interested in. A plasmid vector is a small molecule of DNA that is physically separate from and can replicate independently of chromosome DNA within a cell. Vectors are an agent of moving genetic material and expressing particular genes or make a copy of these genes. A liposome, on the other hand, is an artificially prepared vessel composed of a lipid bilayer. A viral vector, of which if any of you have had a cold in the last few months, as many of us have, will understand to some degree how these works. Viruses can take a piece of DNA and incorporate it into another organism. Pronuclear injection is injection of genetic material into the nucleus of a fertilized oocyte or female gameocyte or germ cell. This is a way of putting the DNA directly where you want it to be placed. The protoplast, or more commonly referred to as stomatic fusion, is where genetic modification of a plant occurs by two distinct species of plants that are fused together to form what we call a new hybrid plant. This hybrid plant will have the characteristics of both. A stomatic hybrid is another word for this bringing together of genetic information. And the ballistic DNA injection is a device for injecting cells with genetic information in it. These are just some of the vectors listed. As you can see, this is an important and growing part of the technologies. So, how does transgenics work? Well, the concepts are that all living organisms carry the same form of genetic code, as we know DNA, and this is made up of small building blocks in organisms. These are base pairs A, C, T and G. And the processes behind this was covered extensively in the previous lectures. This means that a specific DNA sequence will code for the same protein in all organisms. The transgenics in principle is a piece of DNA is cut from one organism and put into the other organism. The figure on the screen describes visually the differences between conventional breeding and transgenics. So let us explore how transgenics is used in animals. The first stage is to identify a trait and a gene that encodes for that trait. Once you have got the trait identified, you can then isolate the DNA, amplify or make many copies of that DNA. Then your objective is to put that DNA into an animal of interest. This is quite a complicated process in animals, much more simpler in plants. The transgene, which is the gene that encodes the trait of interest, is injected into the egg of an animal. This egg with the injected transgene is then implanted into a surrogate. The surrogate grows the baby is delivered, then the offspring, and only the offspring, will hopefully exhibit the trait of interest. Transgenics in animals is simple. It is perhaps the technical details underlining these processes and technology that is perhaps complicated. Now we will explore the process of transgenics in plants. Again, the concepts are the same. It is perhaps the execution or how you go about the methodology that is quite different. First of all, you identify your trait of interest. You then find what genes encode your trait. Once you have identified the gene or genes of interest, your next step is to get them into the plant. To do this, we use a vector. In the lecture on genetics and plants, we did introduce this concept to you. And this slide is a reminder, and perhaps more detail to that reminder, to enable you to understand this technology. In this example, we will use Agrobacterium as the vector. If you remember from the genetics lecture on plants, Agrobacterium is a natural disease that occurs that infects plants. We will place our DNA of interest into the plasmid. Our DNA of interest is referred to as the tDNA, or the transfer DNA. The plasmid is what occurs in the agrobacterium. Once the gene has been inserted into the plasmid, with the use of something called a restriction enzyme and DNA ligase, you can think of simply as these are the processes and or the glue. The DNA then undergoes what we call a recombinant T1 plasmid, i.e. it's ready to insert. In stage 2, your recombinant Ti plasmid is introduced into the plant cells. This occurs by the natural infection vector the agrobacterium has. The tDNA, or your DNA of interest, carries new genes within the plant chromosomes. Once your DNA is in your plant chromosomes, this can be expressed throughout the plant and or to the next generation, depending on the trait and how it is expressed and when it is expressed.
In the scientific articles, you can now read about some technologies that are being developed. These include the zinc finger nucleases, the transcription activator-like element nucleases, and thirdly, a new delivery system by agroinoculation using different specialized vector systems. All of these technologies are being developed in plant genetic engineering. We will not cover these in any detail, as they are in the very early stages of development and not yet available for commercialization. This does, however, give you an idea for how rapidly this field of technology is developing. Molecular biology plays an essential role in all breeding technologies. Here are a couple of examples of what is called high-throughput DNA marker platforms. This means that many DNA sequences can be identified at once or in one run. These technologies include SNPs, microsatellites, AFLPs, and mobile genetic elements. In the past, the identification of 1 or 2 genes was conducted at a time. This has been very expensive and time-consuming. However, technology is gaining pace, and now a new suite of technologies exists so that we can identify many genes or even the whole genome at once. This field is called the science of genomics, or genomics.
I'd like to talk now briefly about data analysis. When you undertake a whole genome profiling, or profiling of a large number of genes, the amount of data that is created, regardless of the technology or technique that you use, is absolutely vast. In the early days, it was very difficult for scientists to analyze this data, and in fact, brand new strategies had to be evolved. Some of the problems with this data is that there are a low number of biological reps, with a large number of information that pertains to each rep. Well, biomathematicians and biostatisticians have been working on this problem for several years, and one of the outcomes of a lot of this research is the technique to apply something called Bayesian machine learning strategies to this data in order to give you data that makes sense. On the left-hand side of the screen, you will note an image that represents a number of sequence genes. This data is then put into the complex Bayesian algorithms, of which the center of the screen shows you just some of the algorithms and how they're connected. Please note, this is just a snapshot of this technology, and there is much more to the complexity of the maths than what is shown on the screen. The data is then represented as outputs that can be easily inferred or interpreted. The introduction of Bayesian machine learning strategies to understand complex breeding traits has made a significant advancement in this field of technologies. It is important to appreciate why this technology has been enabled, particularly in recent years. A lot of that has to do with the cost of sequencing genes. If you looked at how much it cost about 13 years ago to sequence a gene, you needed to put aside about $30 million US. Today, that process can take about 10 minutes. It can cost you $100, and it is predicted to again drop in price and increase in speed. This has made stepwise differences in the ability for us to conduct breeding programs and to advance this technology.
So what are some of the technological difficulties in the GM technology? Well, one such limitation is the DNA addition only, so no DNA replacement. This is still far from gene targeting in crop plants. Secondly, there is no targeted integration in the genome, with possible unintended effects on the plant phenotype. The EU risk assessment has shown that this needs to be addressed. 3. Non-targeted integration with unpredictable effects of transgenome expression or positive effects. Agronomic testing and EU risk assessment procedures are addressing this point. 4. There is a low efficiency in transformations, which may require selection markers or antibiotics resistance, raising safety concerns. Alternatives to the use and the persistence of antibiotic-resistant marker genes in the marketplace of GPMs exist. Extensive risk assessment failed to identify any adverse effects until now. One of the very important components of GMOs and risks associated with it is containing the traits or genetics that you do not want to get out into the environment. This was called controlling gene flow. The molecular farming needs containment. There is little discussion or there is agreement in the scientific community that this is true. Biological containment is an option. This may be achieved by several methods. There is male sterility, seed sterility, cleostomography, inducible promoters of transgene expression, transgenic mitigation, and maternal inheritance. It was said by Johnson and Dolmore in 2002, by choosing the right plant, the development of gene containment mechanisms may be unnecessary.
I'd like to introduce to you now a YouTube video that represents a BBC UK report in 2010 on potato technologies and where they have used GMO biotechnology. This is a very interesting perspective, as it is the perspective of scientists that actually disagree about the advantages of GMO technologies. Please pause the lecture now and listen to the video. While you are listening to the video, please try and answer the following questions. What was the problem the GMO technology was trying to address in potatoes? How would GMOs hopefully correct for this issue? How do scientists produce GMOs? How much money has been spent on the project being discussed in the video? And what are the economic impacts for UK farmers? And what does Patrick Holden, who was the director of the Soil Association at this time, think about the problem? Have a go at answering all of these questions, and once you've answered the questions, then please do continue with the lecture. YouTube video on the use of biotechnologies in UK potato breeding programme. The first question you were asked to address was what was the problem the GMO technology was trying to address? Well, this was the disease potato blight. It is a significant problem in the farming of potatoes both in current day times and in the past. You may remember such incidences in history as the Irish famine or the Irish potato famine. This was indeed caused by potato blight, which was brought on by high levels of moisture. The problem that we currently have is that we can control this disease, but there is a high number of spraying events required, about 15 times a season. This is not only expensive, but has potential impacts on the environment. The second question asked was, how will GMOs correct for this? Well, it is hoped that the correct genes inserted into the crop would enable less spraying of the harmful chemicals. This would have both positive environmental effects and also reduce the amount of chemicals required. This may not be a desirable outcome, however, to the chemical companies. So how do scientists produce GMO potatoes? Well, firstly, they get a wild type potato plants. They identify the traits that exhibit resistance. They do this by a suite of research projects and looking at the literature or what is known currently. They sequence the resistant traits, and once they have the sequence of the resistant traits, they then can insert this sequence of resistance traits into the modern varieties and thus enable disease resistance. Part D sounds quite simple, but as you are beginning to be aware, the technologies of identification of the trait and then subsequent insertion of the trait are indeed complex. The fourth question you were asked to look at was how much money has been spent on the project being discussed in the video. Well, the answer was £1 million. Is that a lot? It would seem that it's not that much when you look for the answer of the fifth question, which was that the UK farmers suffered a loss of approximately £60 million per year. Therefore, a £1 million investment seems like a good return. So Patrick Holden, Director of the Soil Association, thinks there is a problem with this technology. He thinks that a lot of public funds are not producing the public benefits. No farmer benefit threats to the environment and potentially to human health. He may be right. It is difficult to say. Certainly, when you look at the economic input versus the losses, £1 million does seem quite a small amount. However, you have to remember that the public purse for research funding is small and always reduced. Therefore, organisations such as the Soil Association are always competing for these funds. Where they should be best spent is indeed a complex debate. There is no doubt that soil biology and environmental biology is very important, and if you are producing GMOs in the potato trade in part to increase on the environment, then you certainly should be investing money in understanding soil health, particularly that in the concepts of soil communities. Agronomic traits are controlled by genes, but also by gene networks and by heritable and non-heritable gene regulators. Empirical selection based on phenotype is supported by genetic and epigenetic determinants of emerging complexity. We touched on epigenetics last week. This is where one generation has influenced how the genes are expressed in the following generations. Gene regulations, not only gene combinations, must be captured by plant breeding and by selection of plant varieties. These 3 points conclude that of plant genetics and this part of the lecture. A paper that you may want to refer to as its open source is a new paradigm regulatory RNA transcribed from intergenetic non-coding DNA control gene expression or gene silencing. This is one paper on this topic, of which there are many. This will introduce you to the concept of silencing. This is where a piece of DNA or RNA is used to silence or stop the expression of a gene. A long time ago we used to refer to this part of the genome as junk DNA, and that was simply because we did not understand or could not put a function to it. It turns out that some of this silencing or non-coding DNA is very, very important in gene control expression. This is a growing area of our understanding, and certainly a very important one if we are going to imply GMOs into agriculture.
That brings us to the end of part 1 of this lecture. In summary, we have introduced the concepts of genetic engineering, both within this lecture and in the plant and animal breeding lectures. We have given a description of genetic engineering and transgenics. Some terminology in this field is described. Examples of 2 transgenic methods used in both plants and animals are described. We have looked at the development techniques and where the future may lie. We have touched on the introduction to genomics and the role it plays in the production of GMOs. We have looked at an example of genetic engineering of potatoes and the pros and cons of this technology. We've concluded this lecture with some take-home messages about genetic engineering technologies.
Now we are moving to part 2 of this lecture. Please be prepared to stop this lecture at many points, conduct the task or watch the video and then conduct the associated task, and then move on to the next stage. By doing these tasks you will learn about the different components of the technologies. It is only once you have completed the task and inserted the answers of your task will your lecture notes be complete. If you remember, there will be a short test on the topic of genetics, and for preparation of this test it will be assumed that you have watched and read all associated materials in this topic. So let us move on to the first case study. Please follow the following YouTube link. This will give you an example of a list of crops, and I will ask you to complete the following exercise. This YouTube video was published by Cardiff University. I have compiled a number of questions for the take-home messages I would like you to take away from the video. You will find ARG2 FFT Lecture 10 Part 2 Word document with the questions and spaces for you to enter the answers on Moodle. Please stop the video now, download the document and watch the link to this video. You will also find this link directly on Moodle for your convenience. To ensure that you are on the right document, I have amplified question 5, which is to list 6 GMO crops. It is important that you get a good list together so that you understand and can present some examples of GMO crops. The answers to this task will be posted on Moodle later on in the week, but I really would like you to attempt the questions before looking at the answers. The following slide has been used in a previous lecture. I'd like you to have a look at the technology that it is presenting and describe both the name of the technology and what you think is happening in each stage. You will see each stage with an image that summarizes up the process. Have a go at trying to answer this process before moving on to the next stage. You can go back to any of your lecture notes and refer for the answers, but the answer will also be posted with the other answers midweek. The next video I'd like you to watch is about the USDA and its roles in regulating GMOs. You may notice some bias in this video. Please pay attention to this. Even though the bias does exist, it does contain some interesting information about how the USA model regulates GMOs. When you think that most GMOs are farmed in America, it is important to understand this regulation. There is similar regulation here in Australia. One of the most commonly cited examples of GMO agriculture is indeed that of Dolly the sheep. The YouTube link, which is part of Task 4, can lead you to the story about Dolly the sheep. This indeed is an interesting story, and this video will show you the technologies will show you details of this particular technology. In about 100 words, I'd like you to summarize how they cloned Dolly the sheep. Could you please watch the summary of the GMO on the next YouTube as well and list their crops? The next case study is a detailed look at biotechnology in China. Please follow the following link and read the section on agricultural biotechnology. This will give you an interesting insight on how China is using GMOs in both its biotechnology and agricultural industries. Answer the question insert here. It is important that you understand all sides of the argument, both ethically and sustainability of GMOs. Please watch the following videos, which do indeed present different perspectives. Look at the statements that are being made. Are they misleading? Are they using non-scientific referencing for these claims? Or are they in fact correct? Make up your own mind, but please do try and understand why they are making the messages they are. What groups are interested in biotechnology and its applications, and why are they interested? Are they led by logical arguments, or are their arguments based on fear, emotion, and emotive? It is always a good skill set to be able to understand why people are sometimes selling the message that they are. Now that you have been presented with these case studies, and you have listened to the first lecture and the other supporting lectures in this topic, I'd like you to take a moment and reflect. Write a summary on what you perceive to be the advantages, the disadvantages, and most importantly, the ethics of transgenetics. Please spend some time reflecting on this. I wanted to mention pollen tube pathway transformation technologies. This is a mixing of pollen with DNA and then injecting that DNA into the meristome and ovaries. There is a paper by Liu and Wang in 1988 if you wish to learn more about these technologies. This technology is sometimes used on a commercial basis, but is seen more in a research basis, and is included here for completeness of your notes. I will not be testing on this part of the lecture. And this brings us to the end of part 2, and in fact the end of this lecture. So now, students, you should have completed a number of exercises that have given you different examples of different genetic engineering applications that is used in agriculture, either in a commercial or in a research way. You should now understand the technology and the arguments presented in each task. Once you have completed the lecture tasks, you will then have completed your notes. You can now prepare for your tutorial assessment short answer test. This will be appearing on the website next week. This brings us to the end of this lecture.